way, I should have already said this, sorry, but make sure you have your book open to page 262, because as I'm talking, I'm going through these pages that I asked you to read. And I'm sort of saying them in my own words, and I'll refer to some of these pictures. So basically, the human body, and especially the nervous system, depends on nerves' ability to communicate with one another and with the rest of the body. And the cell's membrane can undergo some important changes that allow that to be possible. Okay. The next thing to remind ourselves of, we've got this cell membrane. It's got a phospholipid bilayer with hydrophilic heads and hydrophobic tails, just like before. It's got as well lots of proteins studded throughout the membrane. Okay, if I zoom in on this cell membrane, sort of like you see in figure, or like the big box under number 2 in the bottom half of page 262, we start to see this. We start to see the cell membrane, but it has some proteins, like it has for instance, a potassium channel. It has, for instance, a totally differently shaped sodium channel. Pretend like these are 3D hollow tunnels. These are specific to the ions. Okay, only sodium passes through this one. Only potassium passes through this one. Maybe over here there's a calcium channel. Only calcium would pass through this one. And so, if this thing was wide open, and there's no gate, there's no door, there's nothing, ask yourself, what do you think potassium would want to do? Say this is the inside of the cell, intracellular, and say this is the outside of the cell. Based on what I told you up here, I hope you would say that potassium would really love to leave the cell and go down its concentration gradient from area of high concentration to low concentration, from area of high concentration inside the cell to low concentration outside the cell. That's what things naturally want to do. Same goes for sodium over here. I hope you'd realize that because sodium is so high in the extracellular space, it would really prefer and love to come into the cell. And ultimately, if the body just left everything alone, that's what would happen, and everything would equalize perfectly and come to a standstill, which nature loves that. Nature loves balance, but meanwhile, the body wouldn't be able to do any work. So what we have is this really special sodium-potassium pump that basically forces sodium out, and it forces potassium in against the concentration gradients. It's the pump that actually establishes this gradient, and we like it that way because as this pump sends 3 sodium out, it's sending sodium out into a place where there's already lots of it. It's sending potassium inside the cell where there's already lots of it. And because that's against what nature would naturally do, that means it requires some energy. And the cell gets that energy from ATP. Okay. ATP gets broken down in this chemical reaction, and that, kaboom, supplies the energy that this pump needs to work against nature. Okay. So, this pump establishes this gradient that I mentioned. Okay. Next thing you think about is to start to fill some of this detail into the neuron that we're a little more familiar with. We're familiar with a neuron that looks a little like this, with a cell body and a nucleus. Here there are some dendrites. By the way, not some, but hundreds, sometimes thousands, okay, of dendrites. It's a long axon. And then on this end of the nerve, we've got these axon terminals, which are the very ends. Okay. Everything in brown that I've traced here, that's a cell membrane. It's a phospholipid bilayer like this. There are channels, there are hydrophilic heads, hydrophilic tails, the whole 9 yards, all studded throughout this. Okay. These dendrites in this cell body actually get synapses from other neurons. And not just one, but many. One book describes this as a cell body is blanketed with these connections, known as synapses. Blanketed. Page 263 shows us a picture where there's 1, 2, 3, 4, 5 synapses from this neuron. But in reality, many, many more. But to simplify it and to learn about it and think about it, that's why we do that. There are many different types of these channels. And one important type of channel that I want to tell you about is something called a voltage-gated channel. What it means is this gate has a door. A voltage-gated sodium channel has a door that opens in response to changes in the electrical charge. As it stands in this cell, it's more negative inside and it's more positive outside. Just like on the Earth, there's a north and a south pole that are opposite of each other. There are positive and negative charges in the cell and outside the cell that are opposite of each other. So just like these are north and south poles of the Earth or of the compass, they're opposites. Negative and positive are also opposites. So therefore, we say that a cell membrane is polarized. It's polarized. There's a difference in the charge between the inside and the outside. So we say it's polarized. It's much more negative inside here than positive. And in fact, if we were going to make a graph where 0 is right here, the resting charge of inside of a cell is negative. It's down here. And positive is up here. And say this is, negative 30 or so. So anyway, the point is, the word polarized that you'll sometimes see means that opposite charges exist between the inside and the outside of the cell. If you actually were to make the inside of the cell more positive, you can bring it closer to 0 or more in the positive direction on my graph, it's not opposite anymore. That's called becoming depolarized. If you depolarize a cell, that means you move it in the positive direction. Okay. So, sorry, a little battery check. Make sure this stupid thing doesn't die. Okay, so this neuron here, that's more negative on the inside and more positive on the outside. There's actually changes that can happen which would make the inside of the cell more positive. And then all of a sudden, this cell membrane that was polarized is now depolarized. The process is called depolarization, and it happens in these cells. And what happens is there's a stimulus or a signal for voltage-gated sodium channels to open. Say this nerve sends a signal that causes these sodium channels to open. And then, when they open, the internal environment of the cell changes, and it goes from negative towards being more positive. Just like I started out negative over here, and I'm going in a positive direction. So this cell is actually becoming depolarized. Now, we don't depolarize an entire cell all at once like a light switch, but we can have that effect on a little piece of the cell's membrane, a little patch like a quilt. And then that change in voltage can activate voltage-gated sodium channels. And I'm going into quite a bit of detail here because I know there's a lot of you guys who are thirsty for some more and ready for some more. I'll make it clear as we go what exactly will be on the test, but I really want to paint this picture for you. So a patch of this cell's membrane becomes depolarized. It was once very negative in that area inside, now it's a little more positive in this area on the inside. And what can happen next is that change in voltage can open voltage-gated sodium channels. And this wave of depolarization can actually have the same effect on a patch of membrane next door. And then that change in voltage can actually affect voltage-gated channels over here and depolarize that section, that section, that section, this one, that section, this one, that section. And this wave of depolarization actually can spread. It can spread, and if this initial input, this initial signal is strong enough for this message to be carried across the cell body to this region here, and something really special happens. I hate that term, but I mean I sound like I'm teaching a health class where I'm talking about the birds and the bees, and something really special happened. You'll remember this area of the cell is called specifically the axon hillock. And there's really a tremendous concentration of voltage-gated sodium channels in this part of the cell membrane. And if this wave of depolarization spreads and reaches right here, all of a sudden, kaboom, that message is going to shoot down the axon. Okay? And how exactly it does it is shown really nicely on page 265 at the top. You see those red dots, one right after the other. And basically, what we're talking about here is this phenomenon known as an action potential. These are weird words, honestly, and it's a little hard to understand why they're called this, but the definition of an action potential is here. It's on page 263 in the box called number 3. So let's go back and let's just go through this page 263 together and start to bring everything together. So the membrane potential means there's a difference from inside the cell membrane to outside the cell membrane. That difference is called the resting membrane potential, and all activities of nerves begin with a change in that potential, a change in that voltage, and it's a depolarizing effect where the inside of the cell becomes more positive. A typical stimulus produces a temporary localized change in that resting potential. That's when I was telling you about this really first part right here. There's a little temporary depolarization that happens right there. And it moves us from here, say, to here. Then, if that is able to have that effect next door, and bring us up past the level known as a threshold. You know, a threshold is your breaking point. A threshold is the point that if you cross that line, something happens. A threshold is like your mom at the store saying, I'm going to count to 3. 1. Okay. 2. All right, you're asking for 3. And when she crosses her threshold, something happens. It's a backhand. I'm just kidding. But that's what a threshold means. And if this cell gets up past the threshold and it reaches this area of the neuron here called the axon hillock, then something is going to happen. And the something is the initiation of this thing called an action potential. Look in box 3. If the graded potential is sufficiently large, it triggers an action potential in the membrane of the axon. An action potential is an electrical event that involves one location on the membrane, say right here. Once an action potential develops in one location, it can spread along the surface. The way that it spreads is because it opens up voltage-gated sodium channels. And when those honeys open, then sodium can rush into the cell down its concentration gradient. And as positively charged sodium rushes into the cell, it makes the cell more positive on the inside. And it happens there, and then just like a stack of dominoes, it happens next door, next door, next door, next door, next door. And it's traveling in this direction down the axon, down the axon, down the axon, down the axon until it hits an axon terminal. Kaboom. Okay, so number 4 says, typically, synaptic activity involves the release of chemical compounds by this presynaptic cell. Compounds called neurotransmitters can bind to the postsynaptic cell's membrane and affect it somehow. And I'm going to draw a bigger picture. I'm going to zoom in right here, and I'm going to explain that further. Each neuron cell body is carpeted. There it was, carpeted with tens of thousands of axon terminals, and any number of them could be active at a given time. They might have the effect of activating the next cell. They might have the effect of inhibiting it. It all depends on which neurotransmitter is coming from. Okay. If you turn your page, it's gonna remind you on page 264 of some of the things that I just said, and it's gonna show you this wave of depolarization where the cell becomes a little more positive. Okay. And then it's also going to start to explain how a cell resets, but we'll get to that later. I want you to focus in on page 265 at the top, box number 3 in red. When an action potential develops at one location, the change in that membrane potential here triggers an action potential at the adjacent portion next door. And this little picture says an action potential here triggers one here, and so on in a series of steps, and so on and so forth, like dominoes falling in a line until we reach the axon terminal. Okay, and page 266 begins to tell us what we see in the axon terminal. So let me blow it up for you here. This is the presynaptic cell. Pre means before. We're talking about a synapse, which is a point of communication. Say there's another neuron down here. This point of communication is called a synapse, and I'm going to draw it big for you down here. A synapse has a presynaptic cell, and it has a postsynaptic cell. This is a postsynaptic cell. Post means after. This is the synapse. This is the cell before the synapse. This is the cell after the synapse. This is specifically the axon terminal, one of these little buttons or knobs. Axon terminal. This is phospholipid bilayer, cell membrane. There are voltage-gated calcium channels. I want you to remember calcium when you think of an axon terminal. These are voltage-controlled or voltage-gated calcium channels. Okay. Sorry, one second. I'm sorry about that. So there are plenty of voltage-gated sodium channels here in this part of the cell membrane. And I'll tell you why that's important in a second. What you see in the picture over here are these sacs. These are little sacs or pouches that contain neurotransmitter. Neurotransmitter is a chemical. Okay? And neurotransmitter is a chemical that has the ability to bind to something, like a key into a lock. And I'm going to show you what that looks like in a minute. But neurotransmitters are chemicals that can bind to proteins in cell membrane and have an effect. Okay? So we've got a presynaptic cell's membrane, we've got vesicles inside that are filled with the neurotransmitters, and we've got voltage-gated sodium channels here. Okay, there are a lot of other things in here. I'm simplifying it a bit to show us the structures that are important right now. On the postsynaptic cell's membrane, there are channels, there are proteins. Okay, and suppose these are sodium channels. The difference in these between voltage-gated sodium channels up there, these open in response to the binding of a neurotransmitter. These have a sort of a deadbolt lock right here that if you fit a pink neurotransmitter ball into it, say the vesicles look kind of like this. If you were to fit a pink neurotransmitter into that, this channel would open. What happens when sodium channels on a cell open? Remember up here, the concentration gradient that exists? If we suddenly opened a door that allowed sodium to come in, sodium would gladly rush in. It would gladly flow from an area of high concentration outside the cell to a low concentration inside the cell, gladly. And when that happened, then potentially we could have the same thing starting over again what happened up here, just in the postsynaptic cell, where now this cell is becoming depolarized and that depolarization is spreading, and then maybe this one has an effect down the line. Or perhaps this is a muscle cell, and as the muscle cell depolarizes, calcium is released inside this cell, which prompts tropomyosin to expose myosin binding sites and allow that muscle to contract, for instance. Alright, so let's come back up here and tell this story. The action potential is traveling down the axon terminal. It reaches this part of the cell. Here, when this action potential reaches voltage-gated calcium channels, it causes them to open. And calcium can rush into the cell. Long story short, calcium serves the purpose of allowing these big bags of neurotransmitter to attach to the cell membrane and dump their neurotransmitter out into this space between. It's a process called exocytosis. And now these big pink balls, which represent neurotransmitter chemical signal, are released into the space here. The space here is called the synaptic, C-L-E-F-T, cleft. This is the synaptic cleft, and neurotransmitters release into the synaptic cleft. And guess what? This thing can float down here and bind to the ligand-gated sodium channel here. Cause it to open up, and sodium says, party time. I will rush in here. I will depolarize this cell, and the process will continue in this cell this time. Okay, so that's what I want to talk about for now. There's a lot more to it than this, and I said quite a bit just now. A little bit, potentially more than I intended to. No, about what I intended to. I want you guys to digest this stuff tomorrow.